Hello everyone, I am Avisankar Chini, a PhD graduate student in Dr. Mondal's lab, Department of Chemistry and Biochemistry. So today I am going to present my research on discovery of long-noncoding RNAs associated with inflammation in human. So basically there are two major terms we are talking about here. One is the long-noncoding RNAs and another one is inflammation. Long-noncoding RNAs are basically the longer transcript which eventually does not codes for protein, but they are very important in terms of cell signaling, metabolism. The major limitation of long non-coding RNAs research is that most of the time they are found not to be conserved between species to species. That leads us to look for the human specific long non-coding RNAs. Now, let's talk about a few words on inflammation. Inflammation is a pathophysiological condition where any kind of abnormalities in body is just an outcome of that fact. It's like cardiovascular disease, obesity, autoimmune disease, and also the modern day context with the COVID. The way it works is known as the NF-kappa beta signaling pathway. So when there is an infection, it triggers the TLR4. It is a surface receptor, and in downstream it activates the nuclear factor kappa beta, and eventually that goes to nucleus, translocated into nucleus, and starts synthesizing the response molecules, also known as the cytokines and other pro-inflammatory genes. One of the most highly investigated long non-coding RNAs is Hotair. Our lab first reported that lncRNA Hotair is not just a gene silencer, but also it is involved in inflammation in mouse. So keeping that in mind, based on that fact, we investigated non-coding RNAs in human. Our major target was the discovery of novel long non-coding RNAs associated with inflammation in human and immune response, and also their functional characterization. In order to do that, we differentiated human monocyte cells into human macrophages by some chemical treatments that triggered the monocyte to differentiate into macrophage. Then we treated the macrophage with inflammation stimulus, the lipopolysaccharide from E. coli bacteria. Once it is stimulated or inflamed, we extracted the total RNA, and the total RNA was subjected for RNA sequencing and later on the data analysis. After data analysis, we found thousands of different types of genes which are affected by their expression profile during inflammation. We also wanted to validate the RNA sequencing data. We performed expression profiling of those target RNAs, both protein coding and non-coding as well. We were looking to check their level of expression and validate the RNA sequencing data to make it cDNA, and from cDNA you do real-time PCR to check their expression profile, how much RNA was actually synthesized during that event. Two different way verification. One is immunofluorescence staining. We stain the cells, the differentiated cells, with marker protein known as CD68, and also we did perform one round of real-time PCR. So based on our data, we found there are very new, so far not reported, new genes. Their level of expression is coming up during the inflammation process. We sorted them out, and initially we targeted 5 non-coding RNA genes to analyze their expression profile and their impact on inflammation. So we also were very much interested whether the newly found non-coding RNA, whether they are involved in or been regulated in NF-kappa beta pathway. We also tested for that. Based on the RNAseq data, we also designed gene-specific primers to check their expression profile. We tried to block the nuclear factor kappa beta signaling pathway, and what we found, blocking of nuclear factor kappa beta signaling pathway downregulates the pro-inflammatory cytokines as well as the Hotair, as well as those newly found very new genes not reported anywhere yet, long non-coding RNAs 2 and 3. Most interestingly, even after blocking the NF-kappa beta signaling pathway, the 1 is not affected. After the initial studies, we targeted those 3 lncRNAs, long non-coding RNAs, and also we started looking at their mechanistic studies, how they behave, how they interact with other proteins, and also their functional characterization, their potential role in cell signaling pathway that can lead us to discover some mechanism as well as some future diagnosis and therapeutics as well. I want to thank the National Institute of Health, North Texas Genome Center in UTA. Also, I want to thank College of Science and the whole team of Discover for such a beautiful opportunity.